Welcome to today's video on antinuclear antibodies, or ANA. This is one of the most commonly ordered tests in rheumatology, and also one of the most misunderstood. I'm here to walk you through what ANA really is, how it's measured, and what it means, or doesn't mean, for diagnosing autoimmune diseases. Antinuclear antibodies are immune proteins that mistakenly target parts inside the nucleus of our own cells. They can bind to DNA, histones, and other nuclear materials. ANA is a hallmark of several autoimmune conditions, especially lupus, but also Sjogren's syndrome, scleroderma, and mixed connective tissue disease. But here's the catch. ANA can also be found in healthy people, especially older adults. It can even appear temporarily after infections or certain medications. So context is everything. There are two main ways ANA is measured. The first is called indirect immunofluorescence assay, or IFA. This is considered the gold standard. A blood sample is placed on cells, and if ANA is present, it binds to the nucleus. A fluorescent dye is added, and under a microscope, patterns light up. The titer tells us how much ANA is present, and the pattern, like speckled or homogeneous, can offer diagnostic clues. This method is highly sensitive for lupus, but not very specific. The second method is ELISA or multiplex assays. These use specific nuclear proteins to detect targeted autoantibodies. They're faster and easier to automate, but they may miss some forms of ANA that aren't included in the test panel. They tend to be more specific, but less sensitive than IFA. Different ANA patterns can point to different diseases. Homogeneous patterns often suggest lupus. Speckled patterns may be seen in Sjogren's or mixed connective tissue disease. Nucleolar and centromere patterns are linked with types of scleroderma. And cytoplasmic patterns may indicate myositis or autoimmune hepatitis. So how do we interpret an ANA result? First, a positive ANA doesn't confirm an autoimmune disease. Clinical context matters. Second, low titers are common in healthy people. Third, a negative ANA by EFA makes lupus unlikely. And finally, don't repeat ANA testing unnecessarily. ANA is a powerful tool when used wisely. If this video helped clarify things, give it a like, subscribe for more, and drop your questions in the comments. Thanks for watching.